Today we discussed about plus 2 biology chapter 7 human health and diseases. What is health? According to WHO, health is not just the absence of disease. It is the state of physical, mental, psychological and social well-being. Health is maintained by balanced diet, hygiene, that means personal hygiene and environmental hygiene, exercise and yoga. The term disease is the malfunction of the body and body parts. First, classification of disease. Classification of disease. There are two types of diseases. First, infectious disease and second, non-infectious disease. What is infectious disease? It is easily transmitted from one person to another person. But in case of non-infectious disease, it is not transmitted from one person to another. First topic, common diseases in humans. The disease causing organisms are called pathogens. The disease causing organisms are known as pathogens. They may be bacteria, virus, fungi, protozoans and helminths. We already learned about phylum Protozoa and members. They include malarial parasite and some amoebic parasites also. Helminths, phylum helminths, worms, etc. are also included in the category of pathogen. Bacteria, viruses, fungi, protozoans and helminths. All parasites, external or internal parasites, cause harm to the host. External and internal parasites. External parasites living on the body surface of the host. But in case of internal parasites, they inhabit inside the body part of the host. First, we learn about bacterial diseases. Bacterial diseases. We need to learn about typhoid and pneumonia. Both of them are bacterial diseases. First, typhoid. The name of bacterium, that is the pathogen bacterium, Salmonella typhi. Salmonella typhi is the causative organism, that is a bacteria causing typhoid. Contaminated through food and water. Spreading through food and water. It is an infectious disease that is transmitted from one person to another person through food and water. Symptoms of the diseases: sustained fever in between 39 degree to 40 degree centigrade, weakness, stomach pain, constipation, headache, loss of appetite. In severe cases, death may also occur due to intestinal perforations. Vital test. Viral test means antigen antibody reaction test is used for diagnosis of typhoid fever. Second, bacterial disease, pneumonia. The causative organism, bacteria. There are two species, Streptococcus pneumoniae and Haemophilus influenzae. There are two types of causative organisms, bacteria. They are Streptococcus pneumoniae and second, Haemophilus influenzae. The alveoli, that is the air sacs inside the lungs. The alveoli filled with fluid leads to respiratory problems. Symptoms include fever, chill, cough and headache. In extreme cases, lips and fingernails turn blue because of inadequacy of oxygen in blood. Transmission through droplets, sharing of utensils with an infected person. There are two viral diseases. There are two bacterial diseases, typhoid and pneumonia. Then next, viral disease. Common cold. Common viral disease of nose and respiratory passages causing by rhinovirus. The causative virus is named rhinovirus. Symptoms: nasal congestion and discharge, sore throat, hoarseness, cough, headache and tiredness. Transmission through contaminated objects. Next, protozoan disease. Protozoan disease. Malaria. We need to learn about malaria. It is a protozoan disease. There are 4 types of malaria caused by different species of Plasmodium. Causative organism, Plasmodium. There are 4 types of species are recognized. They are Plasmodium, vivax, Plasmodium malariae, Plasmodium ovale and Plasmodium falciparum. This species, Plasmodium falciparum, is dangerous and fatal category. The life cycle of malarial parasite. Life cycle of Plasmodium. Plasmodium is a digenetic parasite. Plasmodium is a digenetic parasite which requires 2 hosts for completion of its life cycle. Man is the primary host and female Anopheles mosquito is the secondary host or vector. Human is the primary host and female Anopheles mosquito is the secondary host. Mosquitoes are known as vectors. What is the difference between carrier and vector? Example of carrier, housefly. Houseflies carry pathogens on the bristles of the body. The pathogens stuck on the surface area of the carrier, But in case of vectors, the pathogen enter into the body of mosquito for completion of their life cycle. That is the difference between carrier and vector. Female Anopheles mosquito injected protozoids. Here is a life cycle picture of malarial parasite. Female Anopheles mosquito injected sporozoids, that is diploid case, to a number of chromosome sporozoids to the human body. Sporozoids travel to liver, then to RBC. RBC ruptured and release toxic substance hemozoin. The toxic substance hemozoin, which is responsible for this toxic substance hemozoin, which is responsible for chill and fever. Gametocytes, that is gametes after meiosis, male and female gametes, that is gametocytes formed in blood cells. They are haploid, a number of chromosomes. If this infected blood is sucked by mosquito, these gametocytes, that is male and female gametes, enter into the intestine of mosquito. So, mosquitoes are known as vectors. Fertilization, that is the fusion of male and female gametes, take place inside the stomach of the female mosquito to form sporozoids. Then mature sporozoids loaded in salivary glands of mosquito. Again these sporozoids enter into fresh human body by mosquito bite. This is the life cycle of Plasmodium. Next, protozoan disease, amoebiasis. Amoeba is a protozoan. There are different species of amoeba, but one species of amoeba cause, that means the name Entamoeba histolytica, that cause the disease amoebiasis. Amoebiasis. Causative organism is Entamoeba histolytica. Amoebiasis, also known as amoebic dysentery. It is a particular type of dysentery. Transmission through contaminated food and water by fecal matters, that is a waste, digestive waste, that means fecal matters, or associated with unhygienic condition. Amoebiasis, also known as amoebic dysentery, transmission through contaminated food and water by fecal matters or associated with unhygienic condition. Symptoms: constipation, abdominal pain and cramps. Cramps means the muscles become in a contracted state, that is the cramps, that not relaxed after contraction of muscles. Stools with excess mucus and blood. Stool is also the excreta with mucus and blood clots. The amoeba make ruptures on the intestinal wall and mucus and blood exuded out through the fecal matter. Entamoeba is a monogenic parasite. They need only human body, that is a host, for completion of its life cycle. Next, Helminth disease, Helminthus. that is worm. Ascariasis. Ascaris lumbricoid ascarias, that is common worm disease that usually seen in children. Ascariasis. The helminth worm parasite known as Ascaris lumbricoides. Transmission through contaminated food and water. Symptoms include internal bleeding, muscular pain, fever, anemia, and blockage of intestinal passage. It is a monogenic parasite. Egg of parasite reach the soil along with the fecus of infected person. Next, helminth disease, elephantiasis. Elephantiasis. It is also known as filariasis. Elephantiasis also known as filariasis. Here we can see the picture of filariasis. Caused by helminth named Wucheria bancrofti and Wucheria malai. There are two species, Wucheria bancrofti and Wucheria malai. Usually these worms affect the lymphatic vessels and cause their inflammation. We already learned about the lymphatic system. There are two types of circulatory systems, blood circulatory system and also lymphatic system. Lymphatic system include lymph vessels, lymph nodes, etc. The worms affects the lymphatic vessels. Lymph already seen in between intercellular spaces, the cellular exuded fluid, and cause their inflammation in lymphatic vessels and intercellular spaces. The affected parts are limbs, genital organs, mammary glands, etc. Here we can see the affected limb part. Result in enlargement of affected part called elephantiasis. It looks like the elephant of the leg of elephant, so this commonly called elephantiasis. Wucheria, the organism, is a digenetic parasite. They need two hosts for completion of its life cycle. Man is the primary host and female Culex mosquito. Female Culex mosquito is the vector. Transmission occurs through mosquito bite. Usually female mosquitoes, only the causative organisms or vectors, because for the development of egg, the female mosquitoes need blood, components of blood. So only female mosquitoes are the vectors. Males usually feed plant juice. Next, disease caused by fungi, fungal disease. Many fungi belong to the genera Microsporum, Trichophytum and epidermophytum are responsible for ringworms. This is the picture of ringworm infection. There are so many species that are responsible for ringworm disease. Ringworm disease. The responsible fungi are Microsporum, Trichophyton, and Epidermophyton. Are responsible for ringworms. There are 3 genera responsible for ringworm disease. Symptoms of ringworm disease: dry scaly lesions on skin, nails and scalp. On head portion, scalpy lesions associated with itching. Heat and moisture help the fungi to grow. Transmission occurs through clothes and comb. Next topic: prevention and control of infectious diseases. Next topic: prevention and control of infectious diseases. First, for waterborne diseases, the controlling of waterborne diseases. Like typhoid and amoebiasis, etc. Parasite practice personal and public hygiene measures. For waterborne diseases like typhoid and amoebiasis, practice personal and public hygiene measures. Personal hygiene measures means consumption of clean drinking water, keep body clean, eat fresh food. Public hygienic measures include proper disposal of waste and excreta, that is fecal matters, periodic cleaning and disinfection of water reservoirs, ponds and tank, cleaning of disinfection and the cleaning of water reservoirs and pools and tank, etc. Airborne disease like common cold, pneumonia, avoid close contact with infected persons, avoid use of belongings of the infected person. That are the 2 preventive measures. For vector-borne disease, in case of vector-borne diseases, examples: the mosquito spreading diseases like malaria, eliminate vectors breeding places. Introduce larvivorous fishes like gambusia in pond. Introduction of larvivorous fishes like gambusia in ponds. They feed larvae of mosquito. Spraying insecticides in ditches and drainage. Use mosquito net to avoid stagnation of water around residential areas. These are the some preventive measures. Next, heading: immunity. What is immunity? What is innate immunity? Etc. etc. There are lots of points we need to learn. Next topic: immunity. What is immunity? Immunity is broadly defined as the capacity of the body to recognize materials as foreign to itself and to neutralize, eliminate or metabolize them with or without injury to its own tissue. Immunity is broadly defined as the definition: the capacity of the body to recognize materials as foreign to itself. It may be microbes, dust, chemicals, etc. are included in foreign materials. The term we used antigen. Foreign materials are termed antigen. There are two types of antigens. The term antigen is denoted with blood grouping, and also in case of immunity, the antigen means foreign materials that introduced into the body. The capacity of the body to recognize materials as foreign to itself and neutralize, eliminate or metabolize the foreign materials with or without injury to its own body cell. Immunology is the study of organization and functioning of immune system. Immunity means protection against diseases. Edward Jenner is called father of immunology. Edward Jenner is called father of immunology. Types of immunity. Next topic, types of immunity. There are two types of immunity, innate immunity and acquired immunity. First, innate immunity. What is innate immunity? It is also termed natural immunity. Innate immunity also termed natural immunity. The resistance to certain infections from birth. The resistance to certain infection from birth is called innate, inborn or natural immunity. It attained through birth. Four types of barriers support innate immunity. Four types of barriers support innate immunity. Barriers that support innate immunity are, there are four supporting barriers. Types of barriers that support innate immunity. First, physical barriers. Physical barriers. Skin mucous coating of epithelial lining of the respiratory, gastrointestinal and urogenital tracts trap the microorganisms. Here the mucous membranes that seen in the respiratory tracts, that is lungs and associated part, gastrointestinal parts, that means digestive system, and urinogenital tracts means kidney and associated parts. Skin mucous coating of epithelial lining of the respiratory, gastrointestinal and urinogenital tract trap the microorganisms. Second barrier, that is physiological barriers. Physiological barriers. Factors like body temperature, pH, that means the acidity, various body secretions prevent microbial growth. Example, acidic HCl in stomach, saliva, tear, etc. are act as physiological barriers. Third, cellular barriers. Cellular barriers. Polymorphonuclear leukocytes. Polymorphonuclear leukocytes, PMNL, such as neutrophils, granular WBC like monocytes and lymphocytes, macrophages, engulf and destroy microbes. They are the variety of WBC cells. The PMNL include neutrophils, granular WBC, that is granulocyte like monocytes and lymphocytes. In case of WBC, there are two types, granulocytes and agranulocytes. And agranulocytes. Neutrophils, basophils and eosinophils. Neutrophils, eosinophils and basophils. Three phils. Then the agranular WBCs like monocytes and lymphocytes, two sides. Monocytes and lymphocytes. These are granulocytes. WBC include granulocytes and agranulocytes. Granulocytes include neutrophils, eosinophils and basophils. The cytoplasm of these three types of WBC cells with granules. In case of monocytes and lymphocytes, two sides, the cytoplasm of these WBC cells without granules in their cytoplasm. So, here we mentioned polymorphonuclear leukocytes, the cellular barriers such as neutrophils, granular WBC like monocytes and lymphocytes. Monocyte, lymphocytes and neutrophils. Neutrophils, monocytes and lymphocytes that include PMNL. They act as cellular barriers. The fourth physical barrier, that is cytokine barriers. Cytokine barriers. Virus infected cells secrete proteins called interferon. Usually the virus attack the cells and they rupture the cells. These ruptured cells secrete certain proteins called interferons. Virus infected cells secrete proteins called interferons, which are non-specific proteins. These interferons are non-specific proteins which prevent virus multiplication. Virus infected cells secrete protein called interferons. These interferons are non-specific proteins which prevent further infection of nearby cells for viral multiplication. Next topic, acquired immunity. We already discussed about innate immunity. Next topic, the second type of immunity, acquired immunity. Acquired immunity. An individual acquires resistance during his lifetime is known as acquired immunity or adaptive immunity. An individual acquires resistance during his lifetime is known as acquired immunity or adaptive immunity. But the innate immunity attained through birth. The acquired immunity is a specific immune response. The acquired immunity is a specific immune response. Antibodies are produced in response to a specific antigen. We already discussed about what is antigen. Antigens are any foreign materials that enter into our bodies termed antigens. If any type of antigen that enter into our body, special antibodies appropriate to the antigens are produced inside our body. So, the acquired immunity is an antibody produced response to a specific antigen. Antigen is any foreign molecules. Antigen. Definition of antigen. Antigen is any foreign molecule that can trigger the specific immune response. Antigen is any foreign molecules that can trigger or produce a specific immune response. It is characterized by memory. The acquired immunity, the character of acquired immunity is memory based. When a pathogen enters, that may be virus, bacteria or any type of pathogen that enter into our body, at first time body produce a response. At the time of first entry of the pathogen, a specific pathogen, body produce a response called primary response. With a low intensity. The primary response is with a low intensity. When same pathogen enter the body for the second time, the immune response very fast. The immune response of second time is very fast and highly intensified. First expression is very minor, but in second stage or second entry time, the response is very fast and highly intensified. This is called secondary response or anamnestic response. Anamnestic response. Second response is known as anamnestic response. It is very fast and highly intensified. B lymphocytes and T lymphocytes. There are two types of lymphocytes that are associated with the immune response that produced by the bone marrow. B lymphocytes and T lymphocytes are produced by bone marrows. B lymphocytes and T lymphocytes helps for primary and secondary immune response. B lymphocytes produce antibodies. The B lymphocytes produce antibodies. Antibodies are proteins. Antigens are also some certain types of proteins. The B lymphocytes produce antibodies, a protein, in response to a specific antigen or that particular antigen. T lymphocytes do not produce antibodies. T lymphocytes do not produce antibodies but help the B cells to produce them. That is the difference between B lymphocyte and T lymphocytes. B lymphocytes produce antibodies against a particular antigen. T lymphocytes do not produce antibodies, but T lymphocytes help the B lymphocyte cells to produce antibodies. That is the difference. Then next term. What is? We already discussed about antigen. Next term, antibody. The molecules which are produced in response to antigen are called antibodies. That is the definition of antibodies. The molecules which produce in response to antigen are called antibodies. Antibodies belong to a class of plasma globulins. Antibodies belong to a class of plasma globulins. Called immunoglobulins. It's also known as immunoglobulins. That is Ig, the short form of immunoglobulin, Ig. The molecules which are produced in response to antigen are called antibodies. Antibodies belong to a class of plasma globulins called immunoglobulins, Ig. There are 5 classes of antibodies. There are 5 classes of antibodies. They are immunoglobulin G, immunoglobulin A, immunoglobulin M, D and immunoglobulin E. There are 5 classes of antibodies. They are G, A, M, D, E. Immunoglobulin G is the most abundant immunoglobulin in men. Immunoglobulin G is the most abundant immunoglobulin in men. Antibodies are produced by specialized cell known as lymphocytes and plasma cells. Antibodies are produced by specialized cells known as lymphocytes and plasma cells. Lymphocytes and plasma cells. Antibodies are produced by specialized cells known as lymphocytes and plasma cells. Antibodies are specific to antigen. That is very important point. Antibodies are specific to antigens. That is, a specific antibody only binds with a particular antigen for destruction. That is, a specific antibody only binds with a particular antigen. Antigen may be any type of pathogens, chemicals, cold, pollen grains, etc. are included in antigen. A specific antibody only binds with a particular antigen. This property is also known as lock and key mechanism. Lock and key mechanism. The specificity of antibody to antigen is known as lock and key mechanism. Next topic, the structure of antibody molecule. This is the structure of antibody molecule. Each molecule, this is one molecule, antibody molecule. Each molecule formed of 4 polypeptide chains. 1, 2, 3, 4. 4 polypeptide chains. 2 long chains called heavy chains. 2 short chains called light chains or L chains. 2 heavy chains and 2 light chains. The 4 chains held together by disulfide bonds. These 4 chains are bind together by disulfide bond. To form Y shape. Each antibody has 2 portions, a stem and 2 prongs. Each antibody has 2 portions, a stem and 2 prongs. The 2 tips of the prongs bind to a specific antigen in a lock and key manner. So, forming an antibody complex. The antibody bind with a specific antigen and to form an antigen-antibody complex. Here the antigens are attached and form an antigen-antibody complex. The antigen is germs or any foreign material that bind with this prong area. The amino acid sequence of stem protein is similar in all antibody. The amino acid sequence of stem portion. This is common for all types of antibodies. The amino acid sequence of this is a protein. So all proteins are made up of different types of amino acids. Antibodies are proteins. The amino acid sequence of stem portion is similar in all type of antibodies belonging to same class. But the prongs show great specificity. But the prongs of each and every antibodies are different each other. The prongs show great specificity. This is the peculiarity of antibodies. Here You can utilize a precise note for your exam purpose. Next point, that is functions of antibodies. Functions of antibodies. There are mainly three functions. The antibody perform three functions. First, agglutination. Second, opsonization. And third, neutralization. First function, agglutination. What is agglutination? Binding with bacteria or virus. The antibodies binding with bacteria and virus. Second, opsonization. Opsonization means phagocytosis, that is engulfing of antigen or bacteria. Opsonization means phagocytosis or engulfing the bacteria and destruct the bacterial material. That means opsonization, engulfing. Third, neutralization. Neutralization. To neutralize the toxin released by bacteria. The bacteria or any type of organisms or antigens enter into our body. The living antigens produce toxins that cause the symptoms of the diseases. The symptoms of the disease produced by the toxin materials that excreted from the bacteria, virus and other living antigens. So, some type of antibodies neutralize the toxins that released by the bacteria. Example, tetanus toxin. The antibodies destroy the antigens or pathogens through three ways, that is agglutination, binding, opsonization, engulfing and neutralization of the toxins that are released by the pathogens. Example, bacteria, tetanus toxin. Next term, functions of immunoglobulin. Immunoglobulins are also the stages of antibody formation. The one step of antibody formation, that is immunoglobulins. Antibodies already produced from different types of immunoglobulins. The matured immunoglobulins may be defined as antibodies. So, the functions of immunoglobulins or antibodies. Next topic, functions. Functions of immunoglobulins or antibody. This is the primitive form of antibody. Antibodies are matured from immunoglobulins. We already discussed the 5 types of immunoglobulins. The function of IgG, stimulation of phagocytes. Stimulation of phagocytes. We already discussed about the phagocytic property of antibody. IgA, the function of IgA, protection from pathogens. Protection from pathogens. The function of IgM, activation of B cells. activation of B cells, function of IgD present on lymphocyte as surface receptors. The IgD present on lymphocytes, one type of agranulocyte WBC cell, that is lymphocyte. We already discussed about lymphocyte. The immunoglobulin D, that is act as a surface receptor on lymphocytes present on lymphocytes, one type of WBC cells, agranulocyte. As surface receptor. Next, the fifth, immunoglobulin E, IgE. The function, mediator in allergic responses. Mediator, IgE is act as a mediator in allergic response. Mediator in allergic responses. What is allergens? Allergens that include pollen grains, dust, cold, chemicals, food, etc. are some types of allergens that trigger allergic response in our body. So, IgE act as a mediator in allergic response in our body. Next topic, type of immune response. Types of immune response. Two types of immune responses. First, humoral immunity and second, cell-mediated immunity. Types of immunity. First, humoral immunity and second type, cell-mediated immunity. First, humoral. Humoral immunity. What is humoral immunity? Immunity mediated by antibodies produced by body fluids. Immunity mediated by antibodies produced in body fluids. Body fluids means humors such as plasma, blood plasma or lymph. We already discussed about what is lymph. Is known as humoral immunity. Immunity mediated by antibodies produced by body fluids or humors such as plasma and lymph is known as humoral immunity. It is also called antibody mediated immunity. Humoral immunity is also known as antibody mediated immune system. Specific antigen stimulate the production of specific antibody. That is, we already discussed the specificity of antibody and antigen. Specific antigen stimulate the production of specific antibody. Second type of immune response, that is cell-mediated immune response. Cell-mediated immune response. The immunity produced by the activated or sensitized lymphocytes called cell-mediated immunity. The immunity, the resisting capacity produced by the activated or sensitized lymphocytes, that is one type of lymphocyte, WBC, is called cell-mediated immunity. The T cells directly attack the pathogens. The T cells directly attack the pathogens. This system also react against transplant and protect the body from cancerous cells. Transplants means organ transplantation and also in case of cancerous cells. The cell-mediated immunity, that is sensitized or activated lymphocyte mediated immunity, the T cells directly attack the pathogens. This system also react against the cell-mediated immunity system react against the organ transplants and protect the body from cancerous cells. Cell-mediated immunity protect the body from cancerous cell development and also try to reject the organ transplant. Transplanted organs are rejected because the Foreign organs are, our body recognized the foreign organs as one type of antigen or foreign material and try to reject the transplanted organs from our body. In this case, the cell mediated immune response is very important. Before the organ transplantation, some certain type of this cell mediated immunity become reduced step by step by introducing certain medicines that is necessary for transplantation. Next, difference between humoral and cell mediated immunity. We discussed about humoral immunity and cell mediated immunity. What is the difference? Humoral immunity transferred from one individual to another through serum. The individual means human being, animals, etc. are included in individual. The humoral immunity transferred from one individual to another through serum, blood serum, the fluid part, without RBC, WBC and platelets. Cell mediated immunity required transfer of lymphocytes. Cell mediated immunity requires transfer of lymphocytes, one type of WBC cells. The difference between humoral and cell mediated immunity. Humoral immunity transferred from one individual to another through serum, but cell mediated immunity requires transfer of lymphocytes. Next topic. Active and passive immunity. Active and passive immunity. What is active and passive immunity? First, active immunity. The antibody production followed by the exposure of an antigen or disease or germ is called active immunity. The antibody production followed by the exposure of an antigen or pathogen is called active immunity. This can be introduced by vaccination. Antibody production followed by the exposure or introduction of an antigen is called active immunity. Antigen may be pathogens or any other foreign materials. This can be introduced by vaccination. So what is vaccine? What is the importance of vaccination and what is the term vaccine? Vaccines are prepared by which that contain weakened or attenuated pathogens. A dose of antigen secondary given after primary dose, that is trigger dose to accelerate the production of large amount of antibody called booster dose. So what is vaccine? Vaccines are prepared which contain weakened or attenuated pathogen. Weakened or attenuated pathogens are included in vaccines for specific diseases. A dose of antigen, that is vaccine, given to second time after the primary dose is known as trigger dose. The second dose is known as trigger dose. The importance of booster dose or trigger dose to accelerate the production of large amount of antibody, that is booster dose. First primary dose and secondary booster dose. The vaccines are introduced in 2 or 3 courses. The first introduction of vaccine for a particular disease, against for particular disease, is known as primary dose. Then after a certain time gap, the secondary dose or booster dose also introduced that accelerate the production of large amount of antibody. Active immunity development by disease such as mumps, measles, chickenpox. We take vaccines against mumps, measles, chickenpox, etc. We take primary and sometimes in booster doses, etc. Next topic, passive immunity. Active and passive immunity. We already discussed about active immunity. Then next topic, passive immunity. What is passive immunity? Ready-made antibodies. In case of active immunity, the antibodies are prepared by our own body against the vaccine or antigen or other organism. But in case of passive immunity, ready-made antibodies are given to protect the body against antigens. It is called passive immunity. In case of passive immunity, the antibodies are prepared outside the body or in laboratories. Ready-made antibodies are given to protect the body against foreign agents or antigens. It is called passive immunity. It is less efficient than active immunity. It is short-lived, at least 3 months validity. The advantages is that it acts immediately. The advantages of passive immunity is it is actively participate within a less time period. Ready-made antibodies are given to protect the body against foreign antigen or agents called passive immunity. It is less efficient than active immunity. It is short-lived, up to 3 months validity. The advantages of passive immunity is it acts immediately without time lagging. Then two types of passive immunity. Two types of passive immunity. First, natural passive immunity. There are two types of passive immunity. First, natural passive immunity. Natural passive immunity. The immunity transferred from mother to child through placenta or colostrum is called natural passive immunity. The immunity transferred from mother to child through placenta or colostrum or first milk is called natural passive immunity. What is colostrum? The first milk that sucked by the infant from the mother's body is called colostrum. This with the immense amount of antibodies or immunoglobulins. Immunoglobulin G is abundant in colostrum. Then what is placenta? We already learned about placenta in our reproductive system. Lesson reproductive system. Placenta is the connecting part between the developing fetus to the mother's body. A particular type of development with blood vessels and nerves through the placenta. Then exchange of nutrition and waste materials occurs in between fetal body and mother's blood. Next, artificial passive immunity. First we discussed about natural passive immunity through colostrum and placenta. Second, artificial passive immunity. What is artificial passive immunity? Transfer of immunity from an immunized person to a non-immunized person by transferring antibodies. Transfer of immunity from an immunized person, an immunity produced person, an immunized produced person to a non-immunized person by transferring antibodies. That means antibodies already developed in one person and these antibodies are taken and introduced in other person's body. That is artificial passive immunity. Antibodies prepared by injecting pathogens into body of horse and sheep or etc. In commercial purpose, the antibodies are produced by the introduction of pathogen into certain type of animals, vertebrates like horse, sheep, monkeys etc. and the antibodies for that particular antigen or pathogen is developed inside the body of such animals and these antibodies are extracted and purified. Then these antibodies are injected to human body. That is artificial passive immunity. The transfer of immunity from an immunized person, persons means animals, to a non-immunized person by transferring antibodies. That is artificial passive immunity. Next term, what is vaccine and immunization? Vaccine and immunization. Next term, vaccine and immunization. What is vaccine? Vaccine, a preparation of antigenic protein of pathogen or inactivated or weakened pathogen are introduced into the body. A preparation, and vaccine is a preparation of antigenic proteins, that may be antibodies or sometimes the proteins or some chemicals that also included in the category of vaccine. Vaccine is a preparation of antigenic proteins of pathogen, antigenic proteins of pathogen or inactivated or weakened pathogen. In some cases, weakened pathogens are the part of vaccine. In sometimes attenuated pathogens are included in vaccine, or sometimes the protein content of the pathogens are included in vaccines. There are different types of vaccines. Proteins, it may be proteins of antigen or germ, or inactivated or attenuated germs, or maybe weakened pathogens that introduced in our body is known as vaccine. Introduction of vaccine in the body called vaccination. What is vaccine? A preparation of antigenic proteins of pathogen or inactivated pathogen or weakened or attenuated pathogens are introduced in the body. There are different types of vaccines. Introduction of vaccine in the body is called vaccination. The content of vaccine stimulate. The content, that may be proteins, inactivated pathogens or weakened pathogens are introduced that included in vaccine. This vaccine is introduced in our body called vaccination. The content of vaccine stimulate. The content of the vaccine stimulate the production of antibody in human body, help for neutralize the pathogen during actual infection. When proteins, attenuated or weakened pathogens are included in vaccines, such vaccines are introduced in our body for the stimulation of the production of antibodies against that particular antigen. For neutralize the pathogenic activity. In case of chicken pox, if we take example the chicken pox vaccine, which contain the inactive form of chicken pox virus or protein. In case of viruses, proteins are included in vaccine. The protein coat of virus are the component of chickenpox vaccine. If this chickenpox vaccine is introduced in our body take this protein of virus as a antigen or foreign material and produce antibodies against chickenpox virus. That is the process. If the body need a quick response against deadly microbes, that is tetanus. In case of tetanus, body need a quick response, can inject preformed antibodies. So in case of tetanus, preformed antibodies or antitoxins are introduced, because in case of tetanus, body need immediate response against tetanus bacteria. So there is no need of time lag. Introduce already prepared antibodies to the human body for protect against tetanus. If the body need a quick response against quickly or deadly microbes are introduced in our body as antibody or antitoxin. Passive immunization. Next topic, passive immunization. It is the production of passive immunity through the introduction of antitoxin. Antitoxin is a preparation containing antibodies. In case of passive immunization, if the production of passive immunity in our body through the introduction of antitoxins, chemicals. Antitoxin is a preparation containing antibodies. The recombinant DNA technology employed in the production of vaccine preparation. Recombinant DNA technology employed in production of vaccines in bacteria or yeast. Example, hepatitis B vaccine produced from yeast. Recombinant DNA technology employed in the production of vaccines in bacteria or yeast. Example, hepatitis B vaccine produced from yeast. Then once more, the passive immunity. If our body need quick response against certain pathogen or microbe, we introduce antitoxins or antibodies already prepared in our laboratory and introduced directly in our body for quick response. Next term, allergies. Allergies. Allergy means hypersensitivity of our body to a particular foreign material. Hypersensitivity of our body to a foreign material is known as allergy. What is allergy? Abnormal sensitivity or hypersensitivity to any substance is called allergy. Hypersensitivity or abnormal sensitivity of the body to a particular foreign substance is called allergy. The substances which cause allergy called allergens. The substances which cause allergy called allergens. Examples of allergens: pollen grains, dust, cold, heat, sunlight, fabrics, certain food, fur, feathers, drug, different types of drugs like penicillin, etc. cause allergic reactions in certain persons. Antibodies to allergens. Antibodies to allergen combine with the mast cell of the body. If certain type of allergens introduced in our body, the antibodies are produced against these allergens and these antibodies combine with the allergens with the mast cell of the body. First exposure to allergen not cause allergy. On second exposure to break the mast cells. First exposure of allergen not cause allergy, but it trigger our body. On second exposure to that allergen, the mast cells of our body become breakdown, release histamines. Release histamines. The first exposure of allergens not cause allergy but produce some trigger activity. On second exposure to these allergens, the mast cells of our body break down. The mast cells, that is depend upon immunity, mast cells break down and release chemical histamine. These histamines cause inflammatory response in allergic condition. These histamines, that is produced by the breakdown of mast cells, these histamines cause inflammatory response at the time of allergic response. Symptoms of allergy. Symptoms of allergy include inflammation of mucous membrane, running eyes, sneezing, skin rashes, etc. That is also called eosinophils related inflammatory symptoms. The eosinophils, we already discussed about the 3 types of phils. Granulocytes WBC cell. Eosinophil may increase in number at the time of allergic reaction. Allergic diseases, example hay fever, asthma. Examples of allergic diseases: hay fever, asthma, etc. are allergic diseases. What happened at the time of allergy? At the first introduction of allergens, there is no response. But in second exposure of these allergens into our body, the mast cells of our body become breakdown and form or release histamines. These histamines cause inflammatory responses. Inflammation of mucous membrane, running eyes, sneezing, skin rashes, etc. symptoms. Eosinophils may increase at the time of the number or count of eosinophils in our blood increases at the time of allergic reactions. Examples of allergic diseases: hay fever and asthma. In severe allergy case, the victim may die. This is known as anaphylactic shock. Anaphylactic shock. In severe allergy case, the victim or the person may die. This condition is known as anaphylactic shock. Antihistamine drugs suppress the allergic reactions. In medical field, certain types of antihistamine, we already discussed about histamines. The mast cells break down and produce histamines. Antihistamine drugs suppress the allergic reaction. Next topic, autoimmunity. Autoimmunity. What is autoimmunity? The humoral or cell-mediated immune responses. We already discussed about humoral and cell-mediated immune responses. The humoral or cell-mediated immune responses against the body's own tissue. The humoral or cell-mediated immune response against the body's own tissues is called autoimmunity. The immune system mislead and misunderstand, and they thought that our body tissues are the pathogen and act against our tissue system. That is a simple explanation. The humoral or cell-mediated immune response against the body's own tissue is called autoimmunity. Against the actions against body's own tissues. The humoral or cell-mediated immune response against the body's own tissue is called autoimmunity. The immune system recognizes an antigen of its own body. The immune system recognizes or thought our body cells are the antigen and respond against the same or body cells. The immune system recognizes an antigen of its own body. Own body is recognized as an antigen or foreign material, as non-self. The disease associated with autoimmunes are called autoimmune diseases. The diseases associated with autoimmunity is called autoimmune system. Our immune response system recognize or think that our body cells are foreign materials or antigens and act against our body tissue is called autoimmunity. The disease associated with autoimmunity called autoimmune diseases. Then next, immune system in the body. Immune system in the body. The human immune system consists of lymphoid organs, lymphoid cells, tissues and antibodies. The human immune system consists of lymphoid organs, cells, tissues and antibodies. The immune system play a vital role in autoimmune disease. The immune system play a vital role in autoimmune disease, allergic reactions and organ transplantation. The immune system play a vital role in autoimmune diseases, allergic reactions and organ transplantation. We already discussed about that cases. Next, lymphoid organs. Lymphoid organs. The organs where the maturation and proliferation of lymphocytes take place. The lymphoid organs, the organs where the maturation and proliferation of lymphocytes take place. Lymphocytes are matured and differentiated, that is proliferation, within the lymphoid organs. The lymphocytes matured and differentiated within the lymphoid organs. Next topic, two types of lymphoid organs. Two types of lymphoid organs, primary lymphoid organs and secondary lymphoid organs. The primary lymphoid organs, bone marrow and thymus. Primary lymphoid organs are bone marrow and thymus gland. Bone marrows, we already know what is bone marrows. Then the thymus gland, that situated beneath the ribs on the chest area, but it is rudimentary when we matured up to 7 or 8 year old. The thymus gland are more activated in infancy or early childhood stage. The bone marrow and thymus. Next, secondary lymphoid organs. Spleen and lymph nodes, tonsils, pairs, patches, small intestine and appendix are secondary lymphoid organs, where proliferation or differentiation of lymphocytes that associated to immunity take place in response to specific antigen. The B cells and T cells are produced in bone marrow. T cells specifically matured in thymus, and in early stages T cells are also produced in thymus glands. Here T and B lymphocytes are mature and acquire their antigen specific receptors at primary lymphoid organs. Bone marrow and thymus are the primary lymphoid organs. Here T and B lymphocytes mature and acquire their antigen specific receptors. They become activated. The secondary lymphoid organs include spleen, lymph node, tonsils that is situated in neck region, upper neck region, Peyer's patches in the belly region, small intestine, in some certain cells of small intestine and appendix, etc. are included in secondary lymphoid organs. In secondary lymphoid organs where proliferation and differentiation of lymphocytes take place in response to specific antigen. The bone marrow source of blood cells, RBC, WBC and platelets, and the thymus located near heart and beneath the breastbone are a primary lymphoid organ. Thymus gland larger at the time of birth, thus reduced at the time of puberty. That is up to 7 in case of males and up to in 8 case of females. Bone marrow and thymus provide microenvironment for development and maturation of T lymphocytes. The bone marrow and thymus provide microenvironment for development and maturation of T lymphocytes. Lymph nodes. There are lymph nodes. Here we can see the lymph nodes. Lymph nodes are small solid structures. Lymph nodes are small solid structures located at different points along lymphatic system. These are lymphatic systems. The lymph nodes are small solid structures located at different points along lymphatic system. They trap the antigens and microorganisms. The lymph nodes trap the antigens and microorganisms. The lymphoid tissues lining respiratory, digestive and urinogenital tract. The lymphoid tissues usually seen the lining the respiratory, digestive and urinogenital tract are called mucosal associated lymphoid tissues. The lymphoid tissues lining respiratory, lungs, digestive tract, urinogenital tract, kidney and associate part, and reproductive tract. The inner layering or inner tract, inner layering of these organs called mucosal associated lymphoid tissue, MALT, mucosal associated lymphoid tissue. It constitutes 50% of these MALT constitute 50% of lymphoid tissues in human body. Next topic, AIDS. AIDS. What is AIDS? The full form, acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome. The pathogen, HIV, human immunodeficiency virus. Mode of transmission, through sexual intercourse, shared needles, blood transfusion, mother to fetus through placenta. Blood transfusion means the transfer of blood from donor to recipient. Symptoms and signs of AIDS. The symptoms appear in between 5 to 10 years after infection. The symptoms only appear in between 5 to 10 years after the infection of HIV. HIV destroys T lymphocytes, especially helper T cells. HIV destroy the T lymphocytes, especially helper T cells. T cells are help for the production of B lymphocytes. So T lymphocytes are very important. HIV destroys helper T cells of immune system. It leads to loss of immunity, total immunity. The low immune response leads to viral, bacterial and protozoan infection. The total immunity becomes lost and all type of microbes are enter into our body and cause different types of diseases. Unexplained fever, unrecognized fever, diarrhea, loss of weight, cough, night sweat, fatigue, enlargement of lymph glands, etc. are the main symptoms. The treatment, antiretroviral drug. HIV is a retrovirus, that is If we consider the structure of a virus, there is a core material and a protein coat over the core material. In case of HIV, the core material is RNA. So such viruses are known as retrovirus. In case of tobacco mosaic virus, the core material is DNA, but the HIV is retrovirus, the core material is RNA. So the treatment of AIDS include antiretroviral drug, introducing antiretroviral drug as a treatment, but it is partially effective. But these drugs are partially effective. Diagnosis, ELISA test. We can diagnose the presence or absence of HIV, ELISA test. What is ELISA? Enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. ELISA test is usually used for the diagnosis of HIV. Next, replication. Replication of retrovirus. Here, example of HIV. Replication of retrovirus. After enter into the body, the retrovirus enter into our body, virus enter into macrophages. The virus enter into macrophages. Certain type of immune related cell, macrophages, and convert the RNA, its RNA to DNA. The virus, that is a retrovirus, the core material is RNA. After introduced into our body, they enter into macrophages and turned their RNA to DNA and turned their RNA core material to DNA with the help of reverse transcription enzyme. With the help of reverse transcriptase enzyme. Virus enter into our body and then enter into macrophages, the immune related cells, and this virus turn to its RNA to DNA as core material. Core material auto changing of RNA to DNA by utilizing the enzyme reverse transcriptase. The viral DNA, then the core material become DNA, the viral DNA. First the core material RNA. After this process, the reverse transcription process utilizing reverse transcriptase enzyme. The process is known as reverse transcription. The RNA core material become turned to DNA. Then the viral DNA, the virus with viral DNA, take and direct the infected cells to produce more virus particle. Then this DNA of the virus take the control of the cells and directs the infected cells, to produce more virus particles. The RNA virus introduced into macrophages, and after reverse transcription process utilizing this enzyme, the viral core material become turned to DNA. This viral DNA utilize the cell materials and produce more virus particles. The That is a simple explanation. That is, the infected macrophages act like a HIV factory. The infected macrophages act like HIV factory. The HIV attack the T lymphocytes and replicate and produce more virus. Then this next step, the HIV attack the T lymphocytes and replicate and produce more viruses. Then they release into blood and attack other T lymphocytes. This leads to decrease in number of T lymphocytes and patients start to show symptoms and weight loss. Then microbes attack the body like Mycobacterium, that is Mycobacterium tuberculosis, cause tuberculosis. First entering microbe. Then viruses, fungi, parasites like toxoplasma, etc. are introduced into the patient's body. Once more, the virus, retrovirus with the RNA core material enter into macrophages, the immune responsible cells macrophages, and after reverse transcription process by the help of the enzyme reverse transcriptase, the RNA virus become turned to DNA virus, and this DNA virus direct or control the macrophages and produce more DNA viruses within the macrophages. So it act as a, these macrophages act as a factory for HIV. Then next stage, the HIV attack to T lymphocytes and replicate within the T lymphocyte. They replicate and produce more viruses. Then they release into the blood and attack other T lymphocyte. This leads to decrease in number of T lymphocytes and the patient start showing symptoms and weight loss. Then microbes attack the body like then the foreign materials, foreign pathogens like Mycobacterium, viruses, fungi, parasites like toxoplasma, etc. are introduced in our body and cause different types of diseases. Prevention of AIDS. Safe sex. Use of disposable syringes. Educate people by the NACO, National AIDS Control Organization. Facilitate lot of education programs. Next, cancer. Next disease, cancer. What is cancer? It is a dreaded disease characterized by uncontrolled growth of cells. The cell division become altered, resulting in the formation of tumors. It is by the formation of mass of abnormal cell growth and proliferation. There are some certain steps in usual cell division, but this usual cell division become altered and the process become speed up and produce immature huge number of cells. These cell groups are known as tumors. It is a dreaded disease characterized by uncontrolled growth of cell resulting in the formation of tumors through default or unusual cell division. It is by the formation of mass of abnormal cell growth and proliferation. The study of cancer is known as oncology. There are two types of tumors, benign tumor and malignant tumor. Benign tumor and malignant tumor. Malignant tumor. What is benign tumor? The tumor which is confined to the place or stuck on a particular place of its origin called benign tumor. They never proliferate or enter into other places. The benign tumors are formed a particular portion. Usually these are not dangerous. In some cases, some benign tumors may change into malignant tumors or cancer tumors. Malignant tumors means cancerous tumors. The tumor which is carried, malignant tumor, the tumor which is carried by blood and lymph to other part of the body. The parts of the malignant tumor become detached from the original part and they travel through the blood and enter into different parts of the body. Where they multiply and produce secondary growth is called malignant tumor or cancerous tumors. The spread of cancerous cells called metastasis. Next topic, different types of cancer. Different types of cancer. First, carcinoma. First type of cancer, carcinoma. Cancer of epithelial cells. Example, skin cancer, breast cancer, lung cancer. Second type of cancer, sarcoma. Cancer of connective tissue. Example, like bone marrow, bone cancer, cancer of lymph nodes. Third type, melanoma. Cancer of melanocytes. Example, skin cancer. Fourth, leukemia. Cancer of WBC. Example, blood cancer. Fifth, lymphoma. Cancer of lymph nodes and spleen. Sixth, glioma. Cancer of central nervous system, brain and associated part. Next topic, characteristics of cancer cells. Characteristics of cancer cells. What are the characteristics of cancer cells? First, cancer is the uncontrolled proliferation of cells. Uncontrolled development and detachment of cells from its origin. Second, they do not differentiate. Then these maso cells never turn to different types of cells for different purpose. They do not differentiate. They grow and disorganize the tissue in which they occur. They grow and disorganize the tissues in which they occur. They have large nucleus, nucleolus and more ribosomes. They have large nucleus and the nucleolus that is situated inside the nucleus and the protein production centers, ribosomes. They have large nucleus, nucleolus and more ribosomes in these cells. They lose adhesive junctions with other cells. They lose adhesive attaching junctions with other cells. So they are detached from its origin and passing through the blood and fix different parts of the body and again form cancer cells in different parts. Then they are abnormal amount of DNA and hence are genetically abnormal. They are abnormal cells with abnormal amount of DNAs. They have the ability to invade other cells. They have the ability to invade other tissues. They transfer into blood cells and attach it to different parts or tissues and growing. This process is known as metastasis. Then next topic, causes of cancer. Causes of cancer. Carcinogens are agents of cancer. Carcinogens are the agents of cancer. The main carcinogens are UV rays, coal tar, cigarette smoke, mustard gas, asbestos, soot. The carcinogens are UV rays, coal tar, cigarette smoke, mustard gas, asbestos, soot, etc. viruses also associated with a certain type of cancer. Endosulfar, malathion, etc. are also some chemical mutagens. Next, oncogenes and tumor suppressor genes. What is the term oncogenes? What is oncogenes and tumor suppressor genes? Tumor suppressor genes. Oncogenes stimulate cell proliferation. All normal cells with proto-oncogenes. In our body, all cells normally have proto-oncogenes which are involved in normal cell function. The function of proto-oncogene is involved in normal cell division or cell function. Mutation of proto-oncogenes leads to oncogenes. Oncogenes are the producer of cancer cells or oncogenes stimulate cancerous growth. Oncogenes stimulate cell proliferation. All normal cells with proto-oncogenes which are involved in normal cell function. Mutation occur in proto-oncogenes leads to oncogenes. They turn to oncogenes. Many tumor causing viruses carry similar type of genes. This type of viruses also form or produce or induce the formation of cancer cells. Tumor suppressor genes. So tumor suppressor genes suppress tumor formation. Their absence leads to tumor growth. There are two possibilities. First possibility, the mutation occur in proto-oncogenes that turn to oncogenes, and second, the presence of tumor causing viruses. The presence of tumor causing viruses. Third, tumor suppressor genes. The absence of tumor suppressor genes or inactivation of tumor suppressor genes cause tumor formation. Tumor suppressor genes suppress tumor formation. Their absence leads to tumor growth. Symptoms of cancer. Change in bowel or bladder movement. Change in bowel movement, that means the food movement, peristaltic movement, or bladder, movement in bladder or discharging problems. A sore which does not heal. In some cases, the sore which do not heal, that turn to cancer. Unusual bleeding, lump in breast or elsewhere. Indigestion or difficulty in swallowing. Difficulty in swallowing of food. Change in wart or mole. Change the structure of wart and mole. Weight loss. Nagging, cough or hoarseness. Change in bowel or bladder. Excretion and peristaltic movement are altered. A sore which does not heal. Unusual bleeding, lump in breast and elsewhere. Indigestion or difficulty in swallowing of food. Change in wart or mole. The structure of wart and mole become changed. Weight loss, nagging, cough or hoarseness, etc. are some symptoms of cancer. Next, detection and diagnosis of cancer. Detection and diagnosis of cancer. It is based on the characteristics of malignant cells, that is the cancerous cells. WBC count, bone marrow biopsy. What is biopsy? Biopsy is a technique in which a sample of abnormal tissue is removed and tested. WBC count, bone marrow biopsy, X-ray, CT scan, MRI are used for detecting internal organs like kidney and pancreas, etc. Mammography used to detect breast cancer. Histopathological studies of tissue, blood and bone marrow tests considering for leukemia, blood cancer. Antibodies against cancer specific antigens are also used for certain cancer cases. WBC count, bone marrow biopsy. Biopsy is the technique in which a sample of abnormal tissue is removed and tested. In some cases, X-ray, CT scan and MRI are used for detecting internal organs like kidney, pancreas, etc. Mammography used to detect breast cancer. Histopathological studies of tissue, blood and bone marrow tests considering for leukemia or blood cancer. Antibodies against cancer specific antibodies against cancer specific antigens. Antibodies against cancer specific antigens are also used for certain cancer cases. Technique of molecular biology can be applied to detect genes in individuals with inherited susceptibility to certain cancer. Some cancers are genetic, they pass into next generation. So in such cases, techniques in molecular biology can be applied to detect gene in individuals. That means the presence of proto-oncogenes and oncogenes with that inherited susceptibility to certain heritable cancer. Such individuals with heritable chance may be advised to avoid exposure to particular carcinogens, cancer producing materials. Treatment of cancer. Next, treatment. Surgery, radiation therapy and immunotherapy are applicable. Surgery, radiation therapy and immunotherapy. Surgery, removal of tumor mass. Radiation therapy, cancer cells destroyed by using high intensity radiation. Chemotherapy, use anti-cancer drugs orally or direct injection to veins. Hormone therapy, certain type of hormones are used for treatment. Example, estrogen suppress the growth of prostate cancer in males. Estrogen, application of estrogen suppress the growth of prostate cancer in males. Immunotherapy, it involves the augmentation of natural anti-cancer immunological defense mechanism. Immunotherapy is involved like the augmentation of natural anti-cancer immunological defense mechanism. Biological therapy, it involves the application of cancer vaccines to stimulate the immune system. The vaccine application, example using interferon drugs. Next topic, drug and alcohol abuse. Drug and alcohol abuse. Drug and alcohol abuse. The commonly used drugs are, in case of drugs, the commonly used drugs are opioids, cannabinoids and coco-alkaloids. There are three different categories. First, opioids. Opioids are the first category. These drugs bind to specific opioid receptor found in nervous system, that is brain and associated part, and gastrointestinal tracts. In our nervous system and gastrointestinal tract, that is the digestive tract, there are certain opioid receptor centers. These opioid drugs bind to these specific centers. Heroin, that is also known as smack, that is white, odorless, bitter tasting crystalline compound, chemically called diacetyl morphine. This is the chemical structure or bond of morphine. Heroin smack is a white, odorless, bitter tasting crystalline compound chemically called diacetyl morphine. Heroin obtained by acetylation of morphine. Heroin is obtained by acetylation, that is a chemical process of morphine. Morphine extracted from latex of Papaver somniferum. Papaver somniferum. That is poppy plant, common name. Heroin is a depressant. It make depression, mental depression, taken by snorting, that is using through the smoke, and also used by injection. Next category, cannabinoids. Cannabinoids are group of chemicals. Cannabinoids are the group of chemicals obtained from inflorescence, that is bunch of flowers, inflorescence of Cannabis sativa, that is common name Indian hemp plant. Cannabis sativa. Cannabinoids interact with the brain receptors. These cannabinoids, the group of chemicals, interact with certain brain receptor centers. It is also used in Ayurveda and Unani as medicine, small amount. Following narcotics are obtained from cannabis plant. Following narcotics or drugs are obtained from cannabis plant. Bangs are dried leaves, stems and flowering shoots of cannabis plant. Ganja, female inflorescence used as ganja. Charas, that is also known as hashish. Sticky yellow exudation or from leaves, that is the sticky exudation or the just like latex. Sticky yellow exudation from leaves and female inflorescence. Marijuana. Marijuana is a mixture of plants. This cannabis plants or hemp plant, leaves, seeds, stem and flowering tops. Next type of drug, cocaine. That is coca alkaloids. All these chemicals are alkaloids. Cocaine, it is an alkaloid obtained from cocoa plant. The scientific name of cocoa plant is Erythroxylum cocoa. Erythroxylum cocoa. Cocaine interferes with the transport of neurotransmitter dopamine. That the dopamine is a chemical that usually seen in brain. The cocaine is an alkaloid obtained from cocoa plant, Erythroxylum cocoa. Cocaine interferes or interact or mingled with the transport of neurotransmitter dopamine. Dopamine is a chemical that is a neurotransmitter that alter the function of dopamine. Cocaine commonly called coke or crack. cocaine, commonly called coke or crack. It has stimulated central nervous system, that is brain and associate part, producing a sense of euphoria and increasing energy. That increase the energy and they give pleasure. Euphoria and increase energy. Overdose cause hallucination. Hallucination. Illusion. Hallucination means certain type of illusion. Atropa, belladonna and datura also have hallucinogenic properties. Other plants like atropa, belladonna and datura also have hallucinogenic properties. Next type of drug, LSD. Lysergic acid diethyl-amides. LSD full form lysergic acid diethyl-amides. Barbiturates, amphetamines, benzodiazepine, etc. are similar drugs. Some of them are used as medicine. Barbiturates, benzodiazepine, amphetamines, etc. are usually used as drugs. Some of them have medicinal capacity also, used for mental illness like depression, insomnia, loss of sleep, insomnia, depression. Some of the LSD categories are used as medicine. Morphine is a sedative, used in case of operation cases, sedative, and also painkiller. Painkillers have analgesic property. Analgesic property. Then next topic, drug abuse. The unwanted and overdose of drugs impairs one's physical, physiological or psychological function known as drug abuse. The definition of drug abuse: the unwanted and overdose of drugs impairs or alters one's physical, physiological, that is the body functions, or psychological, mental functions, known as drug abuse. Next topic, smoking. Smoking. Tobacco is the dried leaf of plant Nicotiana. Tobacco is the dried leaf of plant Nicotiana. More than 1,000 chemicals in the form of gases. More than 1,000 chemicals in the form of gases. Vapors and particulates. Example, nicotine, carbon monoxide, hydrogen cyanide, phenols, ammonia, formaldehyde, benzene, benzopyrene, tobacco tar, etc. are included in the smoke of tobacco. Next, passive smoking. Passive smoking. What is passive smoking? The intake of tobacco smoke. The inhalation of tobacco smoke by non-smokers. The intake of tobacco smoke by non-smokers from the environmental conditions. Tobacco smoke, that is one of the dangerous situation in case of passive smokers. The smoke enter into our body from their surroundings, also cause same effect of smoking persons. ETS, that means environmental tobacco smoke. It is also dangerous. Passive smoking also dangerous. Harmful effect of nicotine. Nicotine is one of the chemical that present in Nicotiana or tobacco. Nicotine. Lung cancer. Nicotine causes lung cancer. 99% of lung cancer due to smoking. Oral cancer by chewing tobacco. Emphysema. What is emphysema? Alveolar walls, that is the air sacs inside the lungs. Alveolar walls, the wall of alveolus, are damaged, due to which respiratory surface decreases. Emphysema means the damage of the alveolar walls that cause the decreasing surface area. Heart attack. Nicotine increase the production of adrenaline, stress hormone. Stress hormone, adrenaline, Nicotine increase the production of stress hormone adrenaline. It turns increase the heart rate and blood pressure, leads to heart attack. Peptic ulcer, the stomach ulcer, peptic ulcer. Nicotine may increase HCl secretion. The usual pH of HCl in our stomach is 1 to 1.5, that is highly acidic. The adding of nicotine that stimulate the, that is increase the acidic condition, that cause peptic ulcer, the damage of the stomach wall, that is peptic ulcer. The excess acidity damage the stomach wall, that is peptic ulcer. Next topic, adolescence and drug, that is alcohol abuse. Adolescence. Adolescence, drug and alcohol abuse. Adolescence marked by the age between 8 to 18 for girls and 7 to 19 in case of boys. It extends from puberty to complete sexual maturity. The physical changes of adolescence is known as puberty. It is the first external sign of sexual maturation. The change of girls include breast enlargement, hair growth and menstruation, start menstruation cycle. In case of boys, hair growth, chest and shoulders broadens, penis enlargement, voice deepening, facial hair growth, etc. The curiosity, excitement and experimentation drive the youngsters towards drug and alcohol. The stresses like examination, competition, feeling of independence, insecurities, frustrations, depressions, etc. are also associated with this habit. Next term, addiction and dependence. Addiction and dependence. Dependence of the body on drug due to prolonged use is known as drug addiction. Dependence of the body on drug due to prolonged use is known as drug addiction. With repeated use of drugs, the tolerance level of the receptors, the body receptors, we already discussed certain receptors that located in our central nervous system and digestive system. The tolerance level of these receptors in our body increases. With the repeated use of drugs, the tolerance level of receptors in our body increases. Gradually, the receptors respond to the drugs only at higher doses. With the repeated use of drugs, the tolerance level of such receptors in our body increases. Gradually, the such receptors respond to the drugs only at higher doses. This leads to high intake of addiction. Next, withdrawal symptom. What is withdrawal symptom? The drug addicted person find it difficult to discontinue the use of drug. It produces adverse physiological effects which constitute the term withdrawal symptom. Symptoms means a group of discomfort. The drug addicted person find it difficult to discontinue the use of drug. It produces adverse physiological effect which constitute the term withdrawal symptoms. Next, effect of drug and alcohol abuse. Effect of drug and alcohol abuse. refer to the extreme condition in which a person completely addicted to the evil power of alcohol. The term alcoholism refers to the extreme condition in which a person completely addicted to the evil power of alcohol. If the alcohol level above 0.1 percentage in blood, if the alcohol level above 0.1 percentage in blood affects thinking ability, speech movements, reflex, etc., further consumption, further usage leads to blurred vision, impaired vision, incoherent speech, the speech is not clear, loss of body balance. When the level reaches 80 milligram mg per 100 ml of blood, the level of alcohol reaches 80 milligram per 100 ml of blood leads to nausea, vomiting, headache, etc. Then there are other problems that related to alcoholism. The alcoholism may leads to cirrhosis and fatty liver. What is cirrhosis and fatty liver? Where liver cells are replaced by fibrous tissue. The liver cells are damaged and replaced by fibrous tissue. That condition is known as cirrhosis. Fatty liver means the liver become fluidy. The fluid is filled in liver and liver become enlarged. Alcoholic polyneuritis. Alcoholic polyneuritis. Next condition. Alcoholism leads to the water soluble vitamin deficiency. There are two types of vitamins, water soluble and fat soluble. Vitamin A, D, E, K are fat soluble and A and B, C and B complex are water soluble. The alcoholism leads to water soluble vitamin deficiency, B complex deficiency and cause polyneuritis, that damage the peripheral nervous system. Peripheral nervous system. That is the condition polyneuritis. Next problem, heart diseases cause hypertension, high blood pressure and cardiovascular diseases. That is the disorders related to heart and associate part. Amnesia, partial or total memory loss, that is amnesia. The profound consumption of alcohol leads to amnesia, partial or total memory loss. Ulcer and pancreatitis. Ulcer and pancreatitis. Alcoholism damage internal organs. Male impotence. Male impotence. Loss of sexual drive and defective sperm. The structure of sperm become defective and loss of sexual drive. The defective sperm condition is known as necrospermia. Last topic, that is prevention and control. Prevention and control. Avoid undue peer pressure. What is peer groups? The same age group members are known as peer groups. Same age group members are known as peer groups. Avoid undue peer group pressure. Should not compel a child to perform beyond his capacity. Avoid undue peer pressure, that means should not compel a child to perform beyond his abilities or capacities. Education and counseling. Give counseling to face problems, failures, disappointments, etc. Involve sports, reading, music, yoga, etc. are some preventive measures to change their mentality. Seeking help from parents and peer groups. Give proper guidance at the time of need. Give proper guidance at the time of need. So parents and other students can help them. Looking for danger signs. Notice any danger signs they shows. It recognize any symptoms or dangerous signs. Inform to parents and teachers. Seeking medical help. A lot of help is available in the form of qualified psychologists, psychiatrists and de-addiction centers, rehabilitation programs. Now we already complete our this long lesson. If you like this class, please like, share and